it's using. So yeah, so this is the project that we have, and we basically cloned it from the BGWS dataset. We still need to work on the README file because it still has information from the old project. But for today, we're just going to run the test dataset. And before we do that, we need to make a small, what's called, a mini dataset. So let me just go over to the scripts. So we have Comethyl scripts. All right. Yeah. Let's see. All right. There's a script that's missing. That's supposed to basically I want to make a mini dataset that only has chromosome 22. That would help with troubleshooting stuff faster. So let me see, grow all. Which one did I do recently that has a mini data? Scripts. Alright, not this one. Alright, make mini data. Alright. So basically, I'm just going to copy this and add it to our project in the test data set. I'm just going to add make mini data. So basically, this is a Python script that's going to through our cytosine reports, and then that's going to help us only keep 22 and help in generating a mini data set. All right. Yeah, so usually what I do for my scripts is I usually have comments on what it's going to do and then an example of how to use it, or basically how to run it. Does that make sense?
Okay. Yeah.
Yeah. And this is what I've done before, but I just need to make some edits on that are specific to this project. So alright, so Python scripts make mini data. Alright, so input directory. So the input directory that's going to be used for this project is going to be the directory that has your like your full cytosine reports. And for us, it's going to be in the data folder. This is going to be the input directory. So I'm just going to add the full path, and then I think let me just add the full path to the script as well. So usually, if you remember, if you have a path written like this without the forward stroke is a relative path, and the script would only work if you are in the directory that is closest to this dataset. But I'm just adding full path so that it doesn't give us errors. So output directory, I want this to go in the, so we're going to have tests, data, okay. So I'm going to make a data folder, and in this data folder should have the same structure as this one.
Okay.
So it's going to be data folder, and it's going to have 08. Let me just. So data processed, and you're going to add another thing, which is going to be OA status reports. The reason for making sure that they're the same is so that when we're running, so we're going to run code for the test dataset.
Okay.
And then basically, when we're running code for the original thing, we're just gonna be replacing with some paths that are somewhat similar. It will make more sense when when you're actually running the full thing. So yes, this is gonna be our output directory. And then the sample list is supposed to be a list. Alright, so I need to make a text file that has a list of our samples that are going to be used to make the mini data. Let me just try and do that now. So let me just go to the directory with the data. Oh, can I just? Okay, no. So we're gonna ls. So these are the cytosine reports that we need with the CpG dot text, and then we're going to what's called. We're gonna pipe it, and then we're gonna cut. So basically, the sample IDs are the first 4 digits before the underscore, and if you want to grab those, I'm gonna try. If I fail, we'll just use AI. Okay. I'm gonna try and then run the cut. Let's see. No such file or directory. All right. Let's do cut. Cut E. Oh, damn. All right. All right. Let me quickly ask AI for some. I want to. Grab sample IDs separated by first underscore. All right. All right, cool. So this is basically what we want. Let me see what I was doing wrong. So if, oh, okay. I guess I wasn't done. All right, so basically, so all this information is going to be input into a text file, and that's going to be the list of our samples that we're going to use. So that's good. So the text file is going to be in processed. Okay. So I'm going to move this text file a directory up because I just don't want it in my samples. And then I'm going to, okay, I'm going to refresh this. So now we have our text file, and then we're just going to use that as input. So let's see. Alright, so these are the things that we need to run it. If this doesn't work, we'll troubleshoot and see where I'm getting it wrong. Alright, so So yeah, so when it gives you an output like this, it does tell you that it's working. It's going to work on 173 files, and 173 files have been found. And then it's going to be using a filter mode, and it's going to be filtering for chromosome 22. I just selected chromosome 22 because that's the smallest one.
Okay.
And like I think there should be ways of, if you wanted to add like a bed file with different regions, but to keep things simple. And then like the specifics about the Python script you can troubleshoot with AI at your own time. But just wanted to get this. So let's see. So that was a dry run. Let me just double check the other arguments. Okay. So I think I'll just delete the dry run part, and then I should be able to. I think it should. So as it's processing files, it should give an output of what was processed, I believe. But let's just wait and see. All right, I'll open another tab for us to do more work, and then I'll double check this in like 5 minutes. Because it's going through every single file. Okay, all right. So basically, one file has been done.
Out of the 173.
Yeah. So it might take some time for it to completely finish doing everything. So while that's running, what we're going to try and do is we're going to try and what's called fix some of the scripts that we already have and make sure that they're ready to go so that we can do the sample run. Let's see. So all the Comethyl scripts that I have right now are in the Comethyl scripts folder.
Yeah.
So you're gonna have to go in the test dataset. Because right now we're running the test, and the test dataset has a similar structure to like what we were running originally. So yeah, so like I think you opened it. So you this is the second one. Yeah. And then tests, and then scripts. All right. So I'll delete this Comethyl output because this is from a different analysis. Because remember we basically git cloned old different repository just as all the different outputs there. So from our scripts, it's gonna follow the same like steps that I discussed related to Comethyl. I had added a step where you can merge your samples and get a multi-QC file done, but we're going to skip that because we don't have multi-QC for all our samples.
Okay. Yeah.
So our analysis is going to start from 00 import CpG report. So basically, this is just going to be reading in your CpG files.
Okay.
Let's see. Let me just get the scripts ready to run first. Yeah, to save time. So I'll get the scripts ready to run first in this SLURM, what's called Pixie SLURM run. And then at least once that's done, I can see if I can go over some of these scripts, at least what they have, like the comments for the scripts, because going over all of them might take time. I think the goal for now is for you to be able to run it and troubleshoot some stuff. But I guess as you get more advanced, you can be like manipulating the scripts and changing some things if you don't like something.
Oh, great.
If that makes sense.
Yeah.
So this is all right. So let me see. Okay, so it's done 6 files. All right, so this might take some time. So this is, so for running the pipeline, we're going to be using, so you can either set up your environments with Pixie or Conda. We're going to be using Pixie for our reproducible space. And all of these steps are like things that need to be set up so that your Pixie can run. What I'm not clearly sure about is if you're running it, are you able to run it with my file paths, or does that need to be specifically changed to like your file paths? I don't know, but we can double check that. All right, so this is where the Comethyl scripts are starting. Um, Alright, so usually what I do, or what we're going to do is, let's see, so this is for the different project. Right, let me just find, because it's very weird using my mouse on this thing.
Yeah.
And I don't want to make noise. Background. Yeah. Just this. All right. So basically, what we want to do is change these file paths such that they match with this new project that we have. So let's see. All right. So the project root is going to be the main directory in which the project is running. So that's what we're going to start by changing. So the project root for us is, we're just going to right click, copy the path, and then the good thing about Visual Studio Code is you can just highlight something and do Command If, and then when you click this down arrow, you can also do like a replace. So we're just basically going to replace this, so to match the project that we're using. So that's the same thing you're going to have to do basically when you're running like the original data, in that you're just going to have to change the project root, and that project root is just basically going to be like where, I think when you right click on this part, this is the project, because that's where all the data is and all the scripts and the data are.
Right.
So going back to our data, but we obviously need to double check this and see if it's going to work. So because we don't have a merged QC, a merged QC is an output that comes out when you run this script that helps you merge your multi QC and your metadata, but we're not having that. We're just going to have our regular metadata, which is just going to be this one.
Okay.
So let me just check. Do we have so in the test data set, we have a data folder. All right. So I'm just going to copy this over here, and then I'm just going to add it onto here. All right. And then it's going to be. All right. So I'm just going to do Command F to replace this file, Command F, and then that's going to be replaced by this file that has your samples in it. So for the most part, the metadata file needs to be in Excel, because in the script, it only reads in Excel files. So if you have a CSV, you need to convert it to that. Sorry. And then we're also going to change this directory, because we are naming our directory as 08 status and reports. It's not going to be just status and reports. So we're gonna change that as well. Oh, all right. I think I'll just wait on this because let's see. Oh, I can. All right. Or maybe I can rename this one. All right. I'm gonna rename this one to just be cytosine reports, and then once the mini data generation is done, I'm basically just gonna move all the files that we have in the cytosine reports a directory up, and then delete the other directory. Reason for doing it this way is I just want it to be very similar. Like what we have here is the same thing as there.
Okay.
And then that way, when you're now running it on your final data set, the only thing you just need to replace is the project root, and then it'll be ready to go. So that's the thing we need to replace. So I'll change this towards the end, and then all right. So I think yeah. So some of these other folders and outputs that are going to be used as input directory are going to be generated as your script is running. So we don't necessarily need to change anything because these are automatically generated. Does that make sense? Yeah, because like the Comethyl output, right? We don't have a folder called Comethyl output yet.
Yeah.
Because we don't have any outputs yet.
Yeah.
So as we run script 00, we'll start getting those like outputs. Let me just double check and see. Oh. All right, it failed because I renamed this folder as the script was running. I totally forgot that. Yeah, so now it can't read this folder, so because now it's now looking for this.
Okay, you can run it the same thing.
I just need to give it the new. Let me see. All right. Yeah. Let me just do this. All right. All right. So the reason why I'm doing it this way is so that the folder that's being generated across cytosine reports is going to be in here in the process, and then I'm going to just let's see, copy path. All right. Yeah. Okay, print working directory. Okay, so this is the test data. All right, so I'm just going to remove this cytosine report folder.
The one, not the one we just renamed.
Yeah, the old one. Yeah, because it's yes. All right. The reason why I didn't do like the RF because I just wanted what I was doing and delete it. Because if I had just deleted the wrong folder and I was in the data folder, then that would be rerunning this pipeline again. So yeah, those are some things you need to be very careful about, especially when deleting data. So anyway, sorry about that. We're gonna rerun this again.
Okay. Yeah.
This time we just skip the dry run because I know it's working. And then we'll double check on this as it's going. so let me. All right. All right, so I guess I'll go over some of the scripts then, because either way, you still gotta wait for this data to be generated.
Right.
All right, so I'll start off with this first one.
Yeah, skipping the multi-QC for now.
Yeah, so we're gonna skip the multi-QC for now, but okay. If we had multi-QC data on all the samples, we would basically, so let me open the multi-QC folder. So because we don't have multi-QC data for all of them, for all of our samples, we're not going to do this, but basically the multi-QC folder, it should have these folders that have all this information related to alignments, deduplication, and methylation extraction. If it doesn't have them, because sometimes depending with the version of multi-QC that you're using, it might name them differently. But the good thing is the code would tell you what sort of like column names you're trying to identify. So for example, in the alignment, in this text file for alignments, the column names are given like aligned, how many reads aligned uniquely, how many aligned ambiguously, and how many did not align. And then in the code, let me just do, oh sorry, it closed. Let me add a dot so that it doesn't close again. All right, so for example, if you're reading in this multi-QC that's called aligned _.txt, it's going to make sure that you have these column names that are important for the alignment information you need. And then once it has those column names, it's basically going to mutate them and identify the sequencing number. So let me try to find an example of a file that has a sequencing number on it, because all of this information is going to be present. Okay, no, this one doesn't have it, but the raw sequencing files should have the sequencing numbers of when they were actually sequenced. All right, so the sequencing number, is it? All right, sequencing. All right, yeah, so sequencing number is this number that's written as S. So that's like sample 1, 2, 3, 4, 5, 6. For these samples, the sequencing numbers, I think, based on chronological order, they're not really that different. So anyway, the purpose for doing all of this was sort of like so that we get technical variables, some things related to the experiments, so the experiment being when the samples were sequenced. In this code, I didn't include DNA isolation batch, but you could also include it if you had it. So basically, it would basically extract the sample name, the sequencing number. Actually, all right, so this is what it looks like originally, but let me go on to, so yeah, so this text file also has that sequencing number. Okay. So that's basically what it's extracting. And then it's going to filter and group by sample name, and then it's going to sort them by the sequencing number, and then sum across lanes. So because you have multiple lanes, it's just going to add of those different numbers for those multiple lanes to have a final number of how many reads either aligned uniquely or ambiguously. So basically, this script is helping you like go some of those things related to multi-QC. But yeah, we don't have that. So let's see. So this one I explained. And then the deduplication part is also doing the same, just making sure it has the same columns that are important for deduplication. And then once it has those columns, this is just renaming them and then calculating percent duplicated. And then methylation is just extracting the methylation figures, like the percent CpG methylation. So this percent CpG methylation that we're extracting is the same one that is going to be on the MultiQC report. And yeah, as we discussed briefly before, for the MultiQC report, what we look at is for the Bismark percent CpG methylation to be in the same range. And as we hover over these samples, you can see that it's around 80. And then also for the duplicates, we usually get duplicates from the PCR. So those are around the same number. If you had more than 30% or above 40, then consider removing the sample. And then the other thing we look at is percent aligned, which is to do with the mapping efficiency of how well your samples were mapped to the reference genome. And for most of our samples, we don't really need to worry about much. And then I think, yeah, one graph I wanted to show you that I forgot to explain is this one, which is to do with the methylation bias when you're moving from the different positions within your read. It should be uniform. I don't know if you had been there when I presented it in, I think, a lab meeting for Victoria samples. We're using a different method, and then it was like not uniform, basically. And there were some concerns because that would affect the methylation signal you're getting. So the methylation bias should be uniform when you're looking from the first position to the last position. So that's that about the MultiQC. So I guess, yeah, let's see. At the rate at which this is going, we might only discuss scripts today. Or we can, let's see.
Yeah. Well, after this, what else do we need to do to set up the environment?
After this, let me double check that environment is ready to go. I think when I checked yesterday, the environment was ready to go, but let's just make sure. It's just making sure that the Pixie environment is ready to go, and then I think you can do like Pixie run, and then so Pixie test R is Pixie run is basically how you run the Pixie package, and then usually when you're running it, so the way we're going to be running it is going to be Pixie run and then R script. So R script is going to communicate with the R programming language, and it's going to run all different scripts. So Pixie run, in this case, we're just going to test R and make sure it's working. All right, so didn't get any error, so this is fine. So it's telling us it's working. The main important thing to look at is like the version of R that we have. At least that's what I know. Yeah, so this environment is ready. At least I guess what we can also try to do to save time, because is get the other scripts for the original run ready to run.
Okay.
So let's see. So this is the docs, the data folder. Okay. So I'm just going to copy what we have from this test file, the Pixie run test file. And I'm going to add it here. Oh, let's copy the wrong thing. do. Yeah, yeah. Copy. All right. So, we're gonna try and submit a job, or maybe okay. I guess for now we'll just edit the scripts, and then if it's not done, we'll just try and submit some of the jobs for the actual run. At least that will save us some time in the future. But and we hope for the best. Okay. So what we need to do is change the project directory to the one that's going to have our data. So there's something I saw at the top. Okay, so this is so these are some of the, I guess, the commands that you need to look at when you're thinking about what's called when you're using SLURM, which is the job scheduling language that you can use. There's a tutorial about it in the lab GitHub, but I'll just explain at least what we have here. So I'm going to write the job name to be Comethyl Intergen, yeah, CHDS. So when you have the job name like this, when you do your SQ whatever, this is what's going to be listed as. Oh, I wonder if no, I don't have anything running. So these are basically the job names over here. So you remember like when we had snake-make, each rule had like a name,
Yeah.
and such that when you run the snake-make, it will list the name of the job. So when you're using SBATCH, you basically need to just write the name of the thing that you're running.
Mm-hmm.
At least that's what I was told when I started. I don't know what would happen if you had it and run without a job name. Maybe it'll be assigned a random number or something. But and you add in your email. So when you add your email, it'll basically tell you when the job is started and when the job is stopped.
Okay.
I can add your email as well, so that.
Oh yeah, SM Holsinger. Yeah.
Okay. UC Davis. UC Davis. You. And then you'll be getting notifications on whether the job is running or not. And then I guess for mail types, there are different things you can get alerts for. In this case, we set it to all so that you get an alert for when the job has started, when it stopped, when it's been requeued, and I guess there could be other things. So I think you can also do like what's called SBATCH. So SBATCH is the command that you use when you're running or submitting a job. I believe if you do help, you can also get all the information about the other arguments that I'm not explaining. Mm-hmm. Yeah, basically. And then the time is going to be set to 23 hours. So this is your hours are going to be here, and then your minutes, seconds, and then the partition we always set it to high. Number of tasks we always set it to 1, and the number of CPUs I usually change it. So if I'm running, yeah, I think what was I setting it for? All right. So number of CPUs, if you're running it on like your main data, you want it to be as many CPUs as you can. And I think the maximum we have for the genome sync is like 64 per person.
Okay.
So for us, we're gonna write 59. Because sometimes when you have when you have an interactive session like this one, you might open it with like 4 CPUs and so on and so forth. So I just usually set mine to 59. And then the total RAM that I usually set is 700 if I'm running it on the original data. but on the small data set it can be smaller. And then in terms of accounts, you can use LaSalle Lab. You can also use the Genome Center. I don't remember how to spell it on top of my head, but let me try and log in. Oh wait, can you try login on
To the
on the terminal? On the terminal, it's sort of like this then. All right, see when it starts. Yeah. So basically, you just need to write this. Sometimes
the
get
Genome Center group.
yeah Genome Center group, or you can do the public group, and then you see how it tells you how much RAM each user has in terms of limits.
Yeah.
So where it says like a star, it means there's no limit, but where it specifically tells you how much you have. So for the Genome Center, it's like 64 CPUs and 1,024 gigabytes of memory. So I think on the public group, there's also no limit, but I usually, yeah, you can run stuff for more than 3 days in the public group. But anyway, I think most of the time the Genome Center is usually available. And then the outputs or the log files, the standard output and error files. So the standard output is going to be information related to like your run as it's going, and it's running stuff in the background. Everything that's supposed to happen. Like if you were to run the analysis on the terminal, that's the output you'll be getting. But now this output is being saved to a text file called .out. And then if you get any errors or error files, all your errors are going to be there. So these are good for troubleshooting. We'll go over these as we at least running some stuff. Yeah, some of these are defaults. I don't entirely know how to explain. And then this will just have the start date. And then this the dot profile is a hidden file that usually runs in the background that always has the information about like your conda environments and the Pixie and how they're set up. So I added this here to help with our Pixie running, but I don't know. Yeah, we'll double check. I'll try it out, and then you try it out. If it fails with my settings, and then we'll see with your settings, and see if we have to change these in any way. Optional threading hygiene. Okay, I guess these are other commands that are useful for like for threading, which is like the number of CPUs that you're using. These are also some useful commands that are worth looking at. So yeah, this is the other part I wanted to change. So the Pixie workspace is going to be in it's going to be here. All right, it's going to be here, and then the sanity check that's being run is to make sure that your Comethyl is working. All right, actually, let me just run this first as well, because I think there was a time I was getting some errors when I tried to. Okay, so Comethyl is ready to go. Okay, let me see how far this is going. 29 samples. Oh, I wonder if we. Okay, I was considering whether we should just submit a job and add more CPUs, and then it'll be faster.
Oh yeah, instead of 59, something higher.
No, I mean like this is running on this is running based on the settings I have, so it's only using 4 CPUs.
Oh, okay.
So if we submit this script that we have, where's the script? I think okay, yeah. If we were to submit this script as a job,
then it would go a little bit faster.
It might be faster. All right, let me check something. Let me see if we have any free space available to run stuff. All right, let me see the LaSalle Lab. Oh yeah, nice. Okay,
Okay.
we're gonna do an experiment. Where's this? Let's see. Okay, I'm just gonna copy this and then add it to tests, scripts. Okay, let's make mini data. All right, so this is gonna be make mini. All right, wait. So which pixie stuff was I editing? Oh, okay. Oh, okay. All right. All right, cool. No worries. Anyway, let me also add your email as well so that you can see. Let me close some of these. All right, we're gonna change directory, release directory first. Make it go into the data folder. All right, this might save us time. I hope it does. All right, so our lab usually has like 2,000 gigabytes of RAM, and how many CPUs do we have? 128. So because no one else is using anything, we're gonna do like 100.
Okay.
And then so the directory is gonna change. It's gonna be this one. Status of reports. All right.
Okay, yeah. And you just have to run it through the subgroup instead of the genome center group.
Yeah. So basically, what you can do is when you're running jobs or when you're thinking about which one to use, I'll send you the scripts that I have. I guess it's not the script, the command that I have. That I just have in my notes. It'll tell you. So if we do like run this for the genome center, it'll tell you what's running right now in the genome center, and yeah, all these people are using stuff. But sometimes I do like a manual calculation to see if the resources are being all used up to like, because I think the genome center is like 10,000 or 9,856. If you see that the RAM is being used up in totality up to like close to like 9,000, I would go with the public group or the genome center. The problem with the public group is that you're not first preference. It's not first come first. It's just first come first serve basically.
Okay.
And if you use the low group, you can get kicked out.
Oh.
So yeah. So anyway, if the lab stuff is like if the Sol lab is free, that's first preference because that's all lab resource. You don't have to worry about that.
Yeah.
But if it's not, then the genome center is next. If genome center is not working, then
And that's the
public group. Yeah. All right. So experimental script. All right. So we're gonna have. So basically, we're gonna add this script. All right, so I'm gonna delete all this other stuff because this is all stuff that was specific to my Pixie run, and it will waste our time. And we don't need Comethyl now. All right, so we don't need that. All right, so that's just gonna be it, and then it's not gonna be a dry run. It's gonna be an after run. I'm not necessarily worried about the files overriding each other. Does that make sense?
Yeah.
But anyway, so now I'll try submit the job. If it works, in good shape. I believe it should be faster than this, what we're going through. So the way to submit the job, you just type in SBATCH, and then so I basically copied the path of like where the file is.
Yeah.
and then when you do SBATCH, you specify where the file is that you want to run. So the job was submitted.
Did this stop like the old job that we were running?
Which job is that?
Oh, the one that was like going really slow.
It doesn't, because so SBATCH is like using other. I clearly don't understand it myself, but it's using other computational resources, but not on your like interactive shell. Because right now the terminal that we're using is interactive, so 139. Okay, the terminal we're using is interactive, and the outputs we're getting are specific to our terminal. But the SBATCH thing we just run in the background, is at least my understanding. It'll use, it's like a matrix that I haven't clearly understood myself. But anyway, so let me see if our job is running, because I think there might be errors. Okay, we're running. We're going. So you see, like the job as the job ID, the user was using it, the petition, and the name. So our name is Make Mini, and then the time left is 23 hours, and we're using 700 gigabytes of RAM. All right, getting errors. Okay, cool. Let me see. I wonder where my outputs are gonna go, because I've never done this. Like I've never run a job to make a mini data before, so this is very experimental.
Yeah.
So I think the outputs, the mini, what's called, the outputs should all be going into this side of the report file. Let me check what is status reports. Okay, so this is where, like I was trying to find where these files are gonna go, the .out and .error, because they're gonna be telling us information about our run. Okay, that's an hour. Okay, cool. All right, so we'll take a break. Oh, nothing is happening.
Oh, on the job.
Yeah, it's not showing anything. All right, let me go and see in the output folder where it's supposed to go. All right, so we have that. We also have this. And then we're gonna do an ls-alt on it. Okay, so this is supposed to be on job 43. And that's the one that was just made now. Alright, let me just double check if this job is still running. Did you get an email, by the way?
Oh yeah, about the job.
Yeah.
Oh, yes.
So it queued 00 time and it's running. Okay. Okay.
Okay, so that is running, the one that we just submitted, but nothing's happening just yet.
Yeah.
Okay.
Because
is it still waiting? Like
no, it's not in queue because the time has changed. Like if you do an app error and run this again, you see how the time is changing. So it's running, but it's not generating any outputs. I don't know whether I should be concerned or not. But anyway, we'll discuss that after the break. Yeah, let's just take 5 minutes.
Okay.
And then yeah, I'll stop the recording as well.